Alright, so this is going to be a quick revision on biological molecules taken from the lessons, but it's just going to be quick. The take-home message is going to be know which biological molecule has what kind of basic function and the type of test that you will do for that biological molecule. Alright, so the molecules that we're going to talk about are proteins, carbohydrates, and lipids. And then we're just going to look into the examples, the basic structures, and tests for that specific biological molecule. Okay, so the first biological molecule we're going to look into is the proteins. The function of a protein is different. There's different functions. They can be for movement, like the muscles, and this movement is basically a change in shape, which could be a contraction. So you contract and then you have that motion, and the contraction uses ATP. There's just visual of movement, salmon. And you can have enzyme activity from proteins as well. And obviously you can see different proteins, different shape. Here is a more complex 3D shape. This will probably be more fiber. And then, yeah, they all depends on the function. The function and the structure are kind of related in a way, because the function has to, the structure has to permit a specific function to happen. For example, if they had the same shape, then probably you'd require more energy to do something if the shape was kind of not really helping with the function. So that's something important when you talk about proteins, which we'll come to. So the basic structure of the amino acid is shown here. You have your amino group, your carboxyl group, your alpha hydrogen here, which is just showing you that this other group here bound to your carbon is your R group, which varies depending on the type of amino acid that you're talking about. So these different amino acids. You have essential, non-essential. Essential means your body does not synthesize them, like leucine, valine, and you're going to need to provide these to yourself with your diet. And this is in humans. I'm not sure what type of non-essential amino acids you can find in animals, different animals, I'm not sure, but for humans, valine and leucine are your essential amino acids. Okay, so here's just another visual. Carboxyl group, amino group, and then when you have a second one, like this, they react here. The type of bond that you make is the peptide bond, and then as a peptide bond, water is produced as a byproduct of this reaction. The reaction starts with a C. I'm going to let you think about the name of this type of reaction. Yeah. Alright, so your test for proteins is the burette test. That's your reagent for burette reagent. Then you add that. Then your result is a lilac color. If the result doesn't show this lilac color, then there's probably no proteins present in the solution that you're testing for. If you denature the proteins by some kind of reaction, if the proteins are still present, then the color will change. But if you destroy them completely, then it probably won't show. But if you denature them, they might show, because you probably just change the structure of the protein, but it's still protein. But if you, like eggs for example, you fry them, the structure changes. It may become more solid than the semi-liquid state that it was to begin with, but it's still protein. So yeah, just look out for that. Also, that's a very important thing. If you denature a protein because you've changed its structure permanently, or permanently, if you heat it, for example, the structure, the function may change. the enzymes, if you denature them, stop working because the active site has changed shape. So that's something important to remember. In this case, we have primary protein structure, which is just the sequence of amino acid base within that specific protein. So it could be different amino acids. We have beta pleated sheets, which are found in the secondary structure. And these are held together by hydrogen bonds. So your bond that you find in here is a peptide bond. Here are hydrogen bonds. Beta pleated sheets. And your tertiary structure, here you find sulfide bonds, ionic bonds, hydrophilic bonds. You have your quaternary structure, which has more than one polypeptide, so it might be a different polypeptide and another different polypeptide, both 3D. Now they're interacting with each other, making a quaternary structure. So that's important to remember. This is the three-dimensional folding. Within tertiary structure, you take one of these, you take a different type of polypeptide that's folded up like this, bring them together, you have your quaternary structure. Okay. So here's your disulfide bonds, which you find within your cysteine amino acid. So it has to be a specific amino acid. It cannot just be any amino acid. It has to have the sulfur group. Same as the ionic bonds. Hydrogen bonds, hydrogen bonds. Hydrophilic interactions as well. Hydrophobic interactions. It's just types of bonds you find in your tertiary structure. So you can pause here and you can just read more about that. I would definitely recommend you actually read this from your recommended text and do a little bit more reading for yourself from different sources. Carbohydrates. Carbohydrates, they're important to providing energy to move. And this is just showing you how much each organ uses. So this might be energy supplied to move molecules or to use ATP, depends. Obviously this is resting state. When you're thinking or doing complex thought, then maybe the brain uses more than 20%. If you have had too much to drink, alcohol wise, maybe your liver has to break down all that alcohol, probably will increase. When you're running, 10% probably is going to increase. So that's just the basic structure of your carbohydrate for glucose specifically, which you're looking to. That's glucose. You see your carbons, carbon. So that's 1, 2, 3, 4, 5, 6. And counting here is important because we're going to look into the tertiary. We're going to look into the bonding between the carbons, and we're going to talk about 1, 4, 1, 6. So that's why I was just counting that for context. Alright, so what we have here is a glycosidic bond, and same type of reaction. It's going to byproduct. It's going to be water. And again, try to think of the name of this bond, this type of reaction. Okay, so these are simple sugars: glucose, fructose, glucose starch. Okay, so these are complex carbohydrates, which we're going to talk about the structure and function in the slide after testing. Okay, so Benedict's test is the test for your simple sugars, and then depending on the amount, the color change. Obviously, so it's a type of reaction within your simple sugar, so it's going to result in a color change. And the more you have, it's going to be red. If you have moderate, it's going to be orange. Traces is going to be green. Test for starch is the iodine test. There's different tests for. There's a different test for cellulose, but I don't think it's in the spec, in your specific spec. But if it is in your spec and you're just watching this video, then I would recommend you just look that up. Okay, so iodine test is the resulting color is blue-black. If it doesn't change to this color, then and remains this color, then you probably don't have starch in there. Another important thing to recognize is that you can start off with a complex carbohydrate, perfect conditions for enzyme activity, add an enzyme in there, and then slowly it's going to start changing into these colors as you break down the complex carbohydrate into these simple sugars. So if you have a setup where you add an enzyme and it asks you what do you think is going to happen if the conditions are perfect for that enzyme, what you're going to say is the enzyme is probably going to react with the old complex carbohydrate, breaking it down to the simple sugars, and just going to start seeing these colors if the reagent is in the solution. Alright. And what you have is the cellulose. You have this structure here. And then we're going to look at glycogen. We're not going to look at these two. This is like for you to read after I talk about these. So you're just going to look into, okay, we has this structure. What does it do? So that's kind of homework. Cellulose, you see it has these straight structures. Glucose is branched. And you can even see the bonds as well. It's 1, 6, 1, 4. And this one's just 1, 4. So it's just a straight line like that. So glycogen, the function of glycogen is to store energy. So, and energy from carbohydrates is made by the chemoosmosis process in the mitochondria, and this allows ATP to be synthesized by breaking slowly piece by piece. So that's why it's branched. So that's your glycogen. Store energy shaped like this. Cellulose, which is a structure that is used in the plant cell, so it could be an outer wall. It's shaped like this, and you can see it's like, it's kind of like a layer here to protect some from penetration of water or foreign things that you may not want. Depends, but it's micro structure, so it's really tiny, even though it looks big here, but in a micro structure like something huge, will have difficulty penetrating unless it's like tiny. But yeah, it's used as an outer layer. Plants. So you find that in the outer layer. No branches here. So now that we know this is storage of energy and it has branches, just try to figure out, okay, what do these do? So it has branches, so you probably have an idea of what amylopectin does in the plant cell. Alright, lipids. We're not going to look too much into the transport of lipids and all that. That's for later. Right now we're just going to look into the test for the lipids and some basic structures. Not too much. And then finally we're going to look into some functions, structural functions. Pretty interesting, pretty fun. So we have free fatty acids right here. Then we have triglycerides right here. So already know that we find these within the phospholipid membrane, or you can find it within your blood being transported in chylomicrons, but we're not going to talk about that. What we're going to talk about is the emulsion test for lipids. The emulsion test is you add your sample, you add water, you mix them, then you add ethanol, then you mix it, and then if you have a white emulsion, which is shown here poorly by these, it's like mosaic here. You know that you have your lipid within that solution. If you do not observe a milky white emulsion, you probably do not have lipid in there. So it's important to remember the steps. Pour water into your food sample, mix it, then add ethanol. It's important to mix before you add ethanol so that you can see the formation of this emulsion as you add ethanol. Then you can mix it as well, rather than mixing it all at once, because who knows if the solution would have turned before you add ethanol. So it's important to have ethanol just so you can see the extent of that change. Alright, final slide here is just going to talk about the adipose tissue, which is like how you find lipids within your body, the human body, that is. We're not going to talk about beige adipose, just going to talk about white adipose tissue and brown adipose adipose tissue. I apologize for the background noise. Definitely have to improve the setup and hopefully improve the quality of the sound for future purposes. Okay, so for white adipose, you use it to store energy, and so when you are in hibernation or you're fasting, your body is going to tap into these energy stores. It's what people on the keto diet try to do it as a weight loss measure. It's important to consult a physician before experimenting with these kind of techniques, just in case. So that's the storing energy with your white adipose. Brown adipose is used to expand energy and produce heat, and it does this with the same chemoosmosis process that's used by ATP synthase to synthesize ATP in the mitochondria. But in this case, ATP is not being synthesized, so that energy is going into heat to keep someone, like babies have this, so babies, they can't shiver or run around to keep warm when they're still babies, because their muscles are still developing. So they have a lot of brown adipose tissue, and that's why babies just look that's why babies are chunky or fat. I don't know. They have a lot of adipose tissue. Let me say that. And yeah, that helps to keep them warm. Also, I guess wild animals have that hibernate in really cold places. They have a lot of brown adipose tissue to keep them warm as well. Polar bears, perhaps. So that's it. That's the end of this slide compilation of slides video. I don't know. And then it talks about everything that includes the 3 biological molecules: proteins, carbs, and lipids, and the test for them. It briefly talks about their function, but that will be looked into in more detail later. Goodbye.